Okay, so am I audible, Namalita?
Yes, you are audible and visible too.
Okay, thanks. So good evening, one and all. I now welcome all of you to the second session of today's international webinar, which has been jointly organized by IQAC Research Cell, Department of Botany and Department of Zoology. So after the first session, it's my really pleasure to introduce to all of you today's eminent speaker for session 2, Dr. Suchismita Roy. Thank you, Dr. Suchismita, for accepting our invitation and extending your time and efforts to deliver the next session of this webinar. The second session will primarily deal with abiotic stress tolerance in plants. In the last decade, Food and Agriculture Organization, United Nations, has been putting lots of efforts to deal with the major issue of today's world, that is climate change. Well, climate change has become a social, cultural, and scientific taboo for different fields and branches of science and humanities as well. So, putting up climate change data, putting up resilient crops for climate change, and working with all the aspects to combat with the adversities of environment has been a prime focus for all countries and researchers. So there has been a surge in the number of publications which deal with plant growth, plant regulation, productivity, and resilience to climate change through different strategies like phytoremediation, growing up transgenics, improving crop productivity, combating pollution, combating drought, salinity, and different aspects. So before extending my further introduction, today I will shortly introduce our speaker who has already joined the session. So Dr. Suchismita Roy, she is presently a postdoctoral researcher at the University of California, San Diego, at the Center for Cell and Molecular Medicine. She had her educational background from Presidency College, University of Calcutta, followed by her PG from Department of Botany, University of Delhi, and her doctorate from the School of Life Sciences, Jawaharlal Nehru University. Followed by that, she has gained her postdoctoral experience in Sachs School of Pharmacy and Pharmaceutical Science at the University of California, San Diego. She has more than 11 years of experience in cell and molecular biology research, protein-based biochemical assays, biochemistry, and molecular pharmacology. She has rigorously trained herself in GPCR biology and cell-based assays. She has been a visiting graduate student for crystallography studies at the University of Washington. She has also served as a guest associate professor for biochemistry and physiology to undergraduate students at Miranda House, University of Delhi. To her credit, she has several honors and awards from the Council of Scientific and Industrial Research India, Corpus Travel Fund from the School of Life Sciences, JNU, Department of Biotechnology, CTEP International Travel Grant, American Society for Pharmacology and Experimental Therapeutics. She functions as a member of Reviewer Board of Journal of Molecular Sciences, Pharmacology, Experimental Therapeutics, and American Association of Cancer Research. To her credit, she has been working as a reviewer for several international Q1 journals. She has her expertise in optimizing the heterotrimeric protein constructs, validating signaling pathways through microscopy, cellular fractionation, and other techniques. To her credit, she has several publications, and also she has training in different aspects of cell and molecular biology. With these words, now I welcome Dr. Suchismita Roy. Thank you, Dr. Roy, for accepting today's invitation and sharing your precious time. So over to you, Dr. Roy. She will be speaking on the aspects of G protein signaling, abiotic stress response in plants. So over to you, Dr. Roy.
Thank you so much for the invitation from Jangipur College. 75 years of growth is not a matter of small things. So congratulations to the principal, the ex-principals, the current principals, and all the conveners and coordinators for this wonderful one-day webinar. Thank you. A hearty thank you for inviting me to the talk. So to start my talk, is my screen visible and am I audible?
Yes, ma'am.
You can hear everything, right?
Yeah, you're audible and visible. The
Okay.
screen is visible.
Thank you so much for confirming. So I'll be talking about abiotic stress tolerance. So this part of my work was mainly carried out in Jawaharlal Nehru University, where I was a PhD student for quite some time. So, so the topic we are talking about is emphasizing the role of chaperones for understanding abiotic stress response in plants. So chaperones are the kind of proteins which help with what you mean by chaperone is associated protein, which helps in shaping other proteins for better performance. So, going to my main topic. So, don't let our future dry up. So, these are several cutouts from the newspapers and journals which show how worstly we are hit with different kinds of abiotic stress responses. So this is the current map of India, which is generated from CSSRI Karnal. So which shows that how much percentage of the pink region you see, how much sodic salts we have. The yellow region which shows like how much of the area is already under saline stress. So what we see is like with the year number of years increasing, there will be a huge increase in population and decrease in arable land. So we will be left with almost negligible amount of arable land, but a huge amount of uh, mouths to feed on. So we will obviously need a way to strategize our crop production so that we can feed the billions of populations. So in the current rate, you see that we have two kinds of stresses which plants face. One is the abiotic stress and another is the biotic stress. So what is abiotic stress? Biotic stress is any form of stress or strain which the plant faces because of living organism, which can be a fungus, insect, or any microorganisms that it's produce the abiotic stress. And abiotic stress is the one which are generated by natural calamities which include drought condition, heat, cold, submergence, like which happens in flooding stress, and salinity and a heavy metal stress. So in my talk today, I'll be speaking mainly about salinity stress, and it is to be noted that salinity comes with just not one stress. It's a combination stress of two. It goes through the osmotic stress and also a kind of drought stress. So it is the impact of two stress together. So how does it affect the plant? It affects the germination and the growth of the plant, the seeding mortality, like how long the seedling will survive. It reduces the photosynthetic efficiency of the plant. It destroys the capacity of ROS. And ultimately, when we talk about food crops, what we need is biomass. So this kind of abiotic stress also affects the biomass, like the yield of the crop, and ultimately what we need is the crop yield and the kind. So that is also dampened by this kind of abiotic stress forms. So, what are the signaling pathways which get affected with this abiotic stress? So, there are numerous signaling pathways which is working at one go in the plant. So, when a plant is exposed to this kind of abiotic stresses, there leads a immense production of ROS. ROS are ROS is the short form of reactive oxygen species. So reacting oxygen species are generally H2O2, O2. So this kind of species, if liberated from the plant, affects the development and growth of the plant. So we find we find to try like genes from the plants which can help us reduce this ROS activity for the betterment of the plant. So those are called as raw scavengers. Then we have chaperone proteins like the heat shock proteins and the LIA proteins, and the protein which I will talk about today, which is the chaperone. So that is also known as that is a chaperone protein and is called as cyclophilin. So we work on like changes in growth related proteins and other compatible uh, proteins which helps with osmolyte generation and osmotic and ion maintenance. So why I uh, went to the term from gene to protein, because following the central dogma, what it comes to known is like the DNA, RNA, and the protein. So even if we talk about the gene, what finally works in the plant system is like not like plant system, but in any system is the protein system. So the signaling pathways which are affected at different phases are the transcription regulation factors, sensing and perception of the signaling, so that signaling, sensing, and transduction pathways, and activation of a plethora of genes which might be which are stress regulated. So now from where do we get these genes which might be helpful for making stress tolerant plants? So this for about generating abiotic stress tolerant plants, we have devised few methods from where we can find out genes which can be tolerant, which can be helpful in generating tolerant transgenic plants. So what one was the is the regulatory and functional reverse. We can check up the phenotype and then reverse genetics the plant and find the gene which might be responsible for giving abiotic stress tolerance. Second method is by QTL generation. This is often identified by breeding methods, but this method is a very time consuming method. Like it takes around 12 to 15 years to identify a particular QTL and then propagate through breeding programs to identify a gene which might be responsible for providing tolerant plants. Then we have a mutational approach in which we mutate some genes to see if the suppression of that gene is giving us a better tolerant plant. And then another method is by from scavenging genes from the extremophiles. What are extremophiles? Extremophiles are biotic organisms which can survive in high stress tolerant area, like some bacteria grow in high temperature. Some bacteria grows very well in saline conditions. So likewise, there are some plants which can grow in very high saline conditions. So if we can take out genes from those plants and introgress and put them in our normal crop plants, those genes can be used from these plants to make better tolerant abiotic stress tolerant plant. And the last way is from contrasting genotypes or wild relatives. So often we see that for a particular species of plants, there are brothers and sisters of the plants which we don't eat or use it, but they grow it in the wild and they have some traits which makes them more tolerant to particular stress conditions. So that's they are called the contrasting genotypes of plants. So we can take out genes from those plants and put it in our like plant of interest, and we can develop tolerant varieties of plants. So this is also so these contrasting genotypes and extremophile approach is mainly a transgenic form of approach, and others can be brought about by breeding system. So Both are time consuming, raising plants through transgenic approach and then finally propagating in the field and feeding the billion from those crops is a time consuming approach as well as breeding. So it is very important to select the proper kind of gene which will go on for a long time and will be helpful without any side effects is very important. So currently, not all the countries of the world favor transgenic research. Like we can do research, but those crops are not still not edible. So, like India, which doesn't promote that kind of GMO or transgenic research for human consumption, but there are countries like USA, which are very open to genetically modified crops, and most of the corn varieties which are grown in USA, which are different kind of stress tolerant, are GMOs. So some countries have accepted it and some countries have not. European Union is still undergoing the process. Like in countries of Europe, they go through certain validations. If they pass that validation, then they are assumed to be taken up by the uh, uh, people. Like we can use that crop commercially. So, the approach which we have taken to find out a gene for our studies is transcriptome mapping. So we have two, and this transcriptome mapping is by using contrasting cultivars of rice. So there exists a rice variety which is called as the Pokali variety. This variety of rice doesn't produce a beautiful looking rice seed. Like when we consume food, we often look at the kind of seed it produces, like basmati rice. Anybody will be happy to have it because of the long grain, beautiful looking feature, slender whitish look. Everybody will be open to consumption. But pokali is one variety of rice which can grow in high salt condition, but this pokali variety is short grained, fat looking, a little bit of brownish in color. People will not favor is at not like that tasty as I like basmati or IR64 varieties. So this uh, this plant, though it is more tolerant than normal basmati or IR64 varieties of rice, but they have a lesser attention for consumption from the people. So this Pokali variety is found around the coastal region of our country, and we have done a contrasting profiling of this with IR64, and we figured out with real-time and transcriptomic data, we have found out a series of genes which shows upregulation in this category. Like it was more expressed in this wild trait of rice, in the Pokali form of rice than IR64. So this is called as the peptide prolyl cis trans isomerase protein. The other sweet name of this is cyclophilin. Cyclophilin is a chaperone, which I will be talking about today. So what is this cyclophilin? So some proteins tends to have a better arrangement in the cis form, and some proteins have better arrangement in the trans form. Like better arrangement means better activity. So this cyclophilin and like this proteins undergo cis and trans conformation under stress conditions or anything under any kind of stress which is not regular for the plant. So this cyclophilin, which is also called the PPIase or the peptidyl prolyl cis trans isomerase, because of the function of the cis trans conformation, it helps the proteins in restructuring. Like if the protein is favored in the cis form, it tries to keep the protein in the cis form. If that protein is in happy in the trans form, it tries to keep the protein in the trans form. So this gene is particularly helping the other proteins maintain their structure by this activity. And this gene is expressed highly in abiotic stress condition. So uh, this cyclophilin gene acts as a chaperone in different mechanisms of plants, like it helps in maintaining protein stability under stress regulated conditions. They helps in proteins under ubiquitination conditions. They also help in the thylakoid. They also help in other signaling pathway. So the function of this protein is overall in the cell in different forms. So this is a multi-gene family, and in Arabidopsis, it is known to be like we have reported 31 versions of this gene, which gets translated to 41 different kinds of proteins. In rice, it is a gene family of 29 members, but when it comes to after translation, it translates to about 46 proteins. And in Triticum aestivum, since it is a hexaploid plant system, we have this gene around of 212. So it's a huge family of protein, and not all of the members are doing the same function. They have different roles, different localization, like some of them are present in the cytosol, some of them are exclusively present in the chloroplast, some of them are nuclear bound. So this kind of this family is huge, and it has different structures associated with it. So this is a phylogenetic tree, and from here, from the legend key, you can see that it is having different conformations, like some of them have the U-box protein, some of them have the transmembrane domain, WD40 domain. And to be mentioned that this cyclophilin gene also helps in some forms of biotic stress tolerance as well. So first of all, we checked what is the cyclophilin, what cyclophilin is localized in, what is the localization of this gene in our system, like in rice. So the gene which we have specially picked up from this Pokali variety, from now on we will refer to as the OS-CYP2. So this OS means Oryza sativa, which is referring to rice, and CYP2 is the name of the gene, which is called as a cyclophilin 2. So the first experiment which we did is a particle bombardment. Like we make a GFP tag. GFP is the green fluorescent protein, which was tagged to the gene OS-CYP2, and we particle bombarded it into onion epidermal peel to see where it localizes. So where we will see green marks is the place where it is localized. So this is the green area which we showed. It's mainly in the cytosol, and some of them is in the nucleus as well. So this DAPI is showing the nucleus, and this green is showing the nucleus, and this is the cytosolic region, which shows that this protein is present in both the nucleus and the cytosol. So to confirm this, what we did fractionation of the uh, fractionation of cells from Oryza seedlings, and we saw that we see the presence of OS-CYP2 in cytosol and the nucleus. And for markers, so for any experiment to check the exact localization, we use markers. So for the cytosolic marker, we used aldolase, and for the nuclear marker, we used the histone H3 protein. So this shows that it is present in both the nucleus and the cytosol. Now, each and every protein, if it is enzymatic or this kind of chaperone, has enzymatic activity which we can measure. So our next goal was to check whether it is a true cyclophilin or not. So we estimated the protein has this enzymatic activity, which is, I have mentioned that it is called the PPIS activity. So we measured the PPIS activity of this protein, and we found that it is a true cyclophilin because it is matches the enzymatic activity matches with the other proteins, like other cyclophilin proteins of Oryza sativa, zeamase, we show that Oryza sativa is comparative to our gene. So uh, with this, what we got is a true active cyclophilin. So now we checked whether this gene is induced by multiple stress response. For this, we used again the contrasting types of rice. One is the IR64, which is the slender, good-looking human consumable rice, and the Pokali, which is the native genotype, which has less consumption because of the taste, appearance. So this we saw that in case those, so check out this blue marks, which shows that in case of Pokali, this gene was highly regulated, like it has more expression in Pokali. So which validates that that picking up this gene from this wild variety is a good choice for us because we see under different conditions. So this is the salinity stress which I have given. 200 millimolar condition of sodium chloride is often used condition for stress treatment, for salinity stress treatment in rice. And the other conditions which we have given is osmotic stress, heavy metal stress, and high temperature stress. So we see in multitude of stress, the amount of Pokali, like OSIP2 expressed in Pokali is way higher than IR64. So we have actually selected a nice gene from the native cultivar for our work. So before going putting this gene on a edible crop variety, we try to study the expression of this gene in a tobacco plant. So this is a non-consumable plant, but we tested our hypothesis in this easy growing plants to see whether it is performing better or not. So in any physiological experiment in where we are studying abiotic stress, it is very routine to study the shoot length and the root length, germination percentage of this plant, of any plant variety. So we have checked those parameters and we have seen that it performs well. This transgenic tobacco plants perform well in salinity stress and oxidative stress. So now, after working on this tobacco plants, we changed the system and we use the system which is actually required for feeding the millions and the billions. So this is, we tested, we use this gene to produce overexpression plants of rice and also underexpression varieties of transgenic plants. Underexpression, so which we are referring as KD, that is knockdown, and overexpression, which is OE. So we had two lines which will be referred to from now on as OE, which means overexpression of this gene, and KD is the underexpression of this gene. So what we did, we generated this transgenic plants, validated it using different techniques and checking the protein expression, the enzymatic activity, and we saw that this plants does, like the overexpression of this plants does better under salinity stress, under drought condition, and oxidative stress. So what you see in the screen, which is called as a leaf disc assay. So in this assay, we cut small pieces of the rice plant leaves and plate them under the 612 plate and give them different stress treatment. So our hours of stress treatment is mentioned here. It's from 0 hours to 120 hours. And we see how much yellowing is happening under this stress conditions. After that, we collect this and estimate the chlorophyll content. We go for the electrolytic leakage and sodium potassium content. So we see that this overexpression lines did perform better in control conditions in terms of chlorophyll content as well as in salinity, drought, and oxidative stress. And electrolytic leakage and sodium potassium regulation, like amount of sodium or potassium measurement is a routine physiological assays which are done to check the level of tolerance generated by this transgenic plant. So in this kind of assays, we saw that in salinity, the electrolytic leakage means how much the damaged is the membrane. So in salinity stress condition, the overexpression plants had lesser damage to the membranes, so they performed well. Even in drought and oxidative stress, we saw the same condition. So, and sodium potassium imbalance, which is known as the iron homeostasis, how much it is maintained. So, in case of the overexpression lines, it is maintained in a way better form than the knockout lines, like the KD lines. So this is a ultra microscope image, like which is called the electron microscope image. It is called as the EM images. So we see this is on the right hand side. We have the zoomed version of what we see on the left. So under stress condition, we see in the wild type, the thylakoids are broken, like not performing that well. But in you see the overexpression line, the thylakoid is so intact and compact, and but in under expression lines like the KD lines, you see the thylakoids and the granule system, that the photosynthetic system has totally been damaged by salt stress condition. So which confirms that the transgenics which we have raised are having a proper photosynthetic machinery, and that's why that may be one of the reason why this plant is performing better than the wild type or the knockout lines. So in case when we are working on food crops, the main context which we come to is the harvest index and a percentage of field grains. So what is harvest index? A harvest index is the total biomass of the plant by the number of seed it produces, like functional fully filled seeds, like which can be used for human consumption. So, checking out the harvest index and percentage of field grain is very important when we are studying any forms of food crops. So, in this case, we also studied this harvest index and the percentage field grain under this stress treatment. So, first one is the control panel. The second one, we have given a salinity stress treatment. So this is 3 days post recovery, and this is 40 days post recovery. And in the drought condition, this is what happens after this treatment. So we are showing that this under drought condition, the harvest index of this plants was way better. So this is a web diagram showing all the three factors together. This is the control condition, the green one. The blue one is the salinity stress, and the brown one is the drought stress. And we see that overexpression line, especially the OE1, has performed better. And in case of field grains, also we see that the overexpression lines perform better than the knockout lines. So what comes next? We wanted to know whether the antioxidant properties of these plants are high or not. So for like any abiotic stress relation, we mainly study four enzymes. One of is the ascorbate peroxidase, another is the catalase, which helps in the regulations of H2O2, and the other one is the sodium dismutase. So we see in case of sodium dismutase, the difference between these plants under stress and non salinity treated condition was not that much. So under the catalase one, we see in salinity stress it is highly regulated, and in the ascorbate peroxidase it is also high regulated. So MDA is malonaldehyde, which is also one of the stress enzyme indicators, so which also shows that in salinity stress it is quite highly regulated. So this shows that This gene might be affecting the catalytic and the ascorbic peroxidase pathway, but not that much as the sodium dismutase. So, while working, we came through different literatures that auxin is highly regulated by this OSCIP2 gene. So this was when we raised the transgenic, we saw some defects in the root morphology of the transgenics. So here as these two panels are the overexpression ones, and these two panels are the low expression ones. So looking at the root morphology, we thought what might be the uh, causing factor for this. So one of the main hormone which regulates root is auxin. So we went through literature. So we asked the question that whether cyclophilin shows impact has any impact in auxin regulation or not, because we had defect in root morphology. So we saw that there are changes. So depending on the number of lateral roots, they like in the knockout lines, it was highly reduced. Based on the morphology, we tried to check some of the stress responsive auxin regulatory genes, which is IA 29, 11, and 16. These are also transcription factors of auxins. So we saw that they have significant differences in their regulation, which confirms that OSCIP2 might be affecting auxin signaling under non-stress condition. This is a non-stress condition. We have not given salinity stress. This is normal morphology of the plant. So on another hand, when we were trying this protein to uh, crystallize, we had numerous failed attempts. We did not get the proper structure of the protein, which was diffractable. So but we had some clues that this protein might be regulated by another protein, which is another stress responsive regulatory protein, which is called as calmodulin. So calmodulin is a calcium regulated protein which has four arms, and it like regulates calcium imbalance in the cells. Now, how calcium helps in uh, stress treatment? So when there is onset of stress, the calcium ions increases inside in cellular, so the it increases like rapidly to thousandfold. So that is how the cell feels that there is a stress condition, because the content of calcium increases. So we did all these assays in presence of extra calcium. So this one is called the plots which we are seeing. It's called a overlay assay, in which we have conditions when we have treated our blots with more calcium, and we see that this protein does bind to calmodulin. So to validate that, we isolated protoplast from rice and generated the same conditions, and you see with the green mark that wherever you see the colocalization, that it actually interacts with OS calmodulin. But since our crystallographic attempts failed with calmodulin and cyclophilin, we just had to use docking support to see that OS calmodulin, OS is Oryza sativa calmodulin, binds to OS cyclophilin, like Oryza sativa cyclophilin. So the proposed model which we got from our work, and we successfully published it, is like under non-stress condition, OSCIP2 and OS calmodulin might be not interacting together, but OSCIP2 alone is helping in auxin signaling and auxin gene regulations, which is affecting the lateral root formation and seed development. But what happens in the stress with the onset of calcium influx in the cytosol of the cell? OS calmodulin might be binding to OS cyclophilin, and then which in turns affecting uh, a lot of antioxidant genes, and on the other side, it might be affecting the auxin signaling. So this is a small picture of the story which we try to portray for studying the signaling mechanism under abiotic stress in rice plants. So currently, if you want to reach me out, this is my email IDs. And from plant system, while doing a postdoc, I have switched system. Like from plants, I have entirely switched to a mammalian system, and currently I am working with G proteins, and G proteins is, and I am like overwhelmed with this G protein because G protein is a vast protein of science, and nearly 8 Nobel laureates have been awarded for their work on specially G proteins, and 40% of the drug which are available in the market is all are the targets of GPCR and G proteins. So it was an entire switch from my plant system to an animal system. So currently I'm at UC San Diego, and I would like to suggest the students that even with working with the plant background, if I could take a switch from the plant to the mammalian system and get my work published as a in good journals, which shows that yes, if we are open to work, we can work in any system. So lastly, I would like to thank all my mentors, all my schools in which I have worked on, and most importantly, my lab mates. Without this sole survival system, you cannot survive in a lab for so long. And most importantly, all the funding resources. And thank you for the audience.
Thank you, Suchismita. That was a wonderful lecture, and
Thank
it was
you
a
so
very
much.
brief piece of introduction that how we can create some lucrative transgenics to combat salinity. Well, the area of abiotic stress research never ends. It's a dynamic and multi-dimensional field. So there are so many genes and regulators and proteins. So fishing out the right one, which suits best for your plant. That's the most important strategy,
Exactly.
which we feel it's important. Okay, so thanks to you again. And now we will move on to the interactive session. There are some questions from our students and faculties. So Kashish Paria, you can ask the question. So I think she has typed the question. What measures can be taken for rice plants to combat pollution and arsenic issues in our state, that is West Bengal? So Bengal has a lot of arsenic contamination.
Yes, so there are genes which are being studied for heavy metal toxicity. So, like I said, the chart which I showed for all kinds of like how to select genes from different forms, like from the extremophiles or the contrasting varieties, or you can do a breeding screening. So, all these methods can be applied to any kind of abiotic stress. So, currently, there are many genes which have been studied for heavy metal toxicity. Arsenic is one of them, and it is like it is under study, but as you know, India is not an open country for genetically modified crops, so maybe the transgenic approach is not a good way for our country to combat arsenic. So I would suggest that I think pusa and other campuses are working on this kind of heavy metal stress through breeding approaches. Breeding approaches is more time consuming, but that approach has been taken up better in our country. So I think people are working on, I'm sure people are working on arsenic stress tolerance as well.
So there is another question from Rishti Mukherjee. She has thanked you for your informative thoughts, and her question is that uh, the Pokali rice, I think, the economic importance of Pokali rice, it can survive any kind of temperature, or are there any special conditions for its survival?
So Pokali is the contrasting variety which we have used in a study, and we know this variety can resist high salinity because if you go to a rice crop field, you see there is a constant supply of stagnant water. Like there is some amount of water at the base when it is growing, a little bit. So this Pokali variety can constantly take that kind of saline water at their face. Like they can endure that stagnant saline water for their entire growth phase. So, and it grows in coastal region. So I'm sure that as we have tested that it does well in heavy metal stress, high temperature, salinity. So we have genes like from the library screening which we have done. We have other genes also which have performed well in this IR64, which we have taken out from this Pokali variety of rice. So I think Pokali is just because it is not that uh, at a consumable label because it's not good looking. As I said, you won't like to eat a rice which is not slender or not white. That's why it is consumed less. The taste is not that good. That's why people don't favor that kind of rice, and its production is also not that great. So there are options of growing or taking this like other genes from this plant, and it is multi-stress tolerant plant in short.
Okay. So we have a question from Devojit Das. How do chaperones protect from denaturation and improving stress tolerance?
So from my slide, as I said, that chaperone is any protein which helps other proteins. Like the heat shock protein is another kind of chaperone which doesn't work directly in the system, but it regulates the other systems. So in my case, the cyclophilin is a cis-trans isomerase. So isomerase means interconvertible. So this cis-trans isomerase configuration of any protein, like in my case, it might be the calmoleurin, which is regulated by cyclophilin. So a protein might be happy in a cis form or a trans form, depending on in what condition it is. So cyclophilin mainly takes cares of that form. Like if it is in a cis form, it tries to keep the protein in the cis form. If it is in the trans form, it tries to keep it in the trans form. I hope it is clear.
Okay. So we have one more question from Neha Parvin. She wants to know that does the resistant crops have the same amount of nutrient levels as in normal crops?
For my case study, I have checked the amino acid levels, different amino acid level in my plants, and I didn't see any difference. So I can say for my transgenics, the nutrient level did not change, but I have to go through literature to check whether. But yes, there are strategies in which nutrient amount can be increased. Like in my lab, there was another ongoing project in which the lysine amount was increased because lysine is one of the good amino acids which is required for our body. So we were working with lysine as well. So there are ways to increase good amino acids in our rice plants.
Okay. So thank you. We have another question from Shujoy Roy. He wants to know what are the potentialities of plant microbe interaction for stress tolerance.
Yes, there is uh, rhizo-based studies for tolerance. You can inoculate plants with this tolerant varieties and generate tolerant plants. It's true. It's correct. So there
And,
is potential and scope for this kind of studies, plant microbes.
and Shumana Das, she wants to know what are the strategies for developing more tolerance to heat stress, perhaps.
So as I shared in my screen, the five circular things, it goes for all abiotic stress. So if you take a bacteria or a microorganism from a from a hot region and isolate that gene and put it in your plants, maybe you can develop a heat tolerant protein. And the chaperones, like the heat shock proteins or the Lea proteins, these are actually heat stress proteins, as the name says. So these proteins have been extensively for generating heat stress tolerant plants.
Okay. So we have, I think, one question from Mahmood Hassan. He has raised his hand. Yes, please, Mahmood, you can ask.
Ma'am, I want to ask that what are the changes that we can recognize in the nature of the plant that are resistant crops other than the normal crops?
Sorry, I did not get your question. Like, you want to see phenotypic changes?
No, ma'am. Physical changes in the nature of the plant.
Physical, so anything which is drastically different from the normal plants, we won't accept, right? So we will try to generate plants which are similar looking to the variety we eat, like the normal plants. So we will disregard the ones which are showing huge difference in their physical appearance or uh, which is not acceptable, like a super rounded seed of rice. If we generate from your transgenic study, we won't have that, right? So as such, see if we see those traits, we will definitely not encourage growing with that gene. Is my answer okay? Like, did you get my point?
Think so. So
Yes, ma'am. I got it.
there is one more question from Ganapati Basha. He has raised his hand. Yes, please.
Ma'am, first off, thank you for your valuable and informative presentation. Actually, I have two questions. So first, are there any roles of calmodulin proteins in salinity tolerance to plants? And
Yes.
another is, are there any chances of sodium avoidance strategies and reducing uptake?
So there are first to your answer to your first question is calcium binding proteins uh, are like secondary like calciums are like the secondary messengers which amplifies your signal under any kind of stress, be it abiotic or biotic. So regulation of this calmodulin kind of genes is one of the candidates for developing tolerant varieties of any transgenic plants. And can you go to your second question again, please?
Okay, ma'am. Are there any chances of sodium avoidance strategies and reducing uptake?
Yes, so there are many channel proteins which are worked on, or the histidine kinases, which are like regulated by sodium, potassium, or sodium hydrogen influx, efflux. So yes, there are strategies of using these sources for developing tolerant plants affecting sodium regulation.
Okay, thank you, ma'am.
Most welcome.
Okay, so if any one of the participants are having any question, so please ask in the chat box or you can raise your question. So, there are lots of questions coming up, and this discussion is a never-ending topic. It's so much of relevance to physiology and biology.
I have a question. I have
Yes,
a just a
our
general
faculty,
question.
Dr. Suman Karmakar, he has a question.
Thank you, ma'am, for your nice presentation. I have a general query. What is the similarity between the mammalian cyclophilin in related to the plants? And you have studied the different level of expressions of the cyclophilin in plants under different stress conditions like salinity. Have you developed any transgenic plants related to your study?
Yeah, the entire study which I have done is using overexpressing cyclophilin. So I have generated two transgenic. One is overexpressing the gene, and the other is the underexpressing the gene, like knocking out the expression of this gene. So for any validatory studies, the gene has to be overexpressed or it has to be knocked down to study its effect. So I have used both the approaches in my throughout study, which I presented today. And what was your other question?
Now, how the plants cyclophilin structure and something in their relation to the mammalian just cyclophilin.
Oh, it's a very interesting question. So this cyclophilin, which is present in the plants, some of them are chloroplast localized. So those cyclophilins are exclusively found in plants. They are not present in the animals. And when I studied this protein structure of this gene, it has a unique motif which is present only in the plants, not in animals. So there
Okay.
is a difference between plant and animal cyclophilins.
Okay. Thank you. Huh?
Most welcome.
Okay, so if there are any more questions, we can wait for the participants. And the session was really relevant with lots of communications coming up. And rice is really an important staple food crop across the world. And so thank you, Suchismita, once again for your nice presentation. And after the end of session 2, now we have gradually come towards the end of the webinar. Now I will invite our colleague, Ms. Sushmita Sarkar, Assistant Professor in the Department of Zoology, to continue with the valedictory session and to deliver the vote of thanks. So over to you, Sushmita.
Thank you, Shomoda. So finally, good evening, everyone. As we come to the conclusion of today's webinar, I would like to extend my heartfelt gratitude to all of you for your active participation and engagement. This event has been a great success thanks to the contributions of several individuals and groups. First, a special thank you to our keynote speaker, Dr. Preetham Sarkar, Associate Professor, Department of Food Process Engineering, NIT Rourkela, for his invaluable insights into sustainable food packaging systems using agricultural byproducts. We are also deeply grateful to Dr. Suchismita Roy, postdoctoral researcher at the University of California, San Diego, USA, for her enlightening talk on abiotic stress response in plants. I would like to now acknowledge the unwavering support of our governing body members, Mr. Shivashish Chatterjee, President, Dr. Prarthita বিশ্বাস, Principal, Shri Rejaul Karim and Shri Yusuf Hussain, State Government nominees, Dr. Shantinath Sarkar and Dr. Nafisa Banu, University nominees, teacher representatives, Shri Krishnendu Pal Chaudhuri, Shri Torun Mondol, and Dr. Ashish Mondol. Non-teaching representative, Shri Jawaharlal Singh. A special note of thanks to our principal ma'am, Dr. Prarthita বিশ্বাস, for her continuous encouragement and technical support. We are also grateful to our IQAC coordinator, Dr. Basudev Chakraborty, and the entire IQAC team, along with the departments of Zoology and Botany and the research subcommittee. I'd like to extend a special thanks to Dr. Koel Boshu and her team from the seminar subcommittee, as well as Shri Torun Mondol for his role as convener of the documentation subcommittee. A big appreciation to Dr. Nobonita Mukherjee, convener, and Dr. Shommu Mukherjee, joint convener, for their exceptional work in organizing and moderating today's event. Lastly, my sincere thanks to the members of Departments of Botany and Zoology, Dr. Chunki Chaudhuri, Dr. Shumon Kormokar, Mr. Anup Kumar Mondol, Ms. Onkita Mishro, and Mr. Prithvish Shorkar for their dedicated efforts to ensure the smooth running of this webinar. A final word of appreciation goes to all our internal as well as external participants from various colleges, universities, and institutions. Your enthusiasm and insightful queries have added immense value to their sessions. And to our sessions and to our students, your active engagement throughout the webinar has been truly commendable. Thank you all once again for your dedication and contributions. And with these words, I finally conclude today's webinar. Thank you and good night.